talked to Mr. Kizito also.
Yes, I did. So he said he'll give me feedback when he's done making the other papers.
Okay, okay, that's good then. Okay, so today's lecture will will continue from where we ended. We are still looking at these pathways. Okay, so we did the hexose monophosphate shunt, and today I want us to look at the uronic acid pathway. This is another branch of the oxidative pathways that comes from the products of glycolysis and other things. Okay, so we will look at this pathway in details and see what products are produced in this uronic acid pathway. So usually the uronic acid pathway, which is the abbreviated as UAP, this is a uronic acid pathway. So this is a pathway which is also known as the glucuronic pathway. Okay, and in other words, it's another alternative pathway for the oxidative pathway of glucose. So this uronic acid pathway, its products, it produces the uronic acid, and it also produces the pentoses, and it also produces vitamins, which is in most cases vitamin C, which is the ascorbic acid, except in primates and also in guinea pigs. This exemption, we will look at it in details when we look at the pathways, this particular pathway in details, because there is one enzyme that we are going to look at in this pathway, which is absent in guinea pigs and also in humans and these other primates. That's the enzyme that makes this exemption. This is why you find that vitamin C for us humans, usually we need to take it as a supplement. We need to get it from other things, maybe from foodstuffs or as a drug, and etc. etc. Okay, so we will look at this UAP pathway. So the glucuronate molecule, this molecule which is produced in this pathway, it's highly polar. Okay, when we say it's highly polar, it means it's very soluble. Okay, it can dissolve in water very quickly. Okay, that's why it says it's the highly polar. It's a highly polar molecule. So this glucuronate, usually it's incorporated into proteoglycans. Okay, this is a mixture of proteins that makes one compound with glucose and makes with carbohydrates, especially disaccharides, and makes one molecule which are called proteoglycans. Then it also combines with molecules such as bilirubin and also steroid hormones. This glucuronate molecule, it can also be combined with certain drugs, which will make those drugs increase their solubilities. Like I just said, the molecule is highly polar, meaning it has a very strong charge that is attracted to water, and it dissolves, it highly dissolves in water. So when this molecule, the glucuronate, combines with certain drugs, it will increase their solubility, and those drugs can be dissolved in water. So this is just a summary of this pathway that we are going to look at in details. So this pathway usually it comes from the glucose 6-phosphate. Okay, we remember our glucose 6-phosphate, okay, from glycogenesis and also glycolysis. This glucose 6-phosphate, this is where our uronic acid pathway also starts from. Okay, so this glucose 6-phosphate, it's acted upon by this enzyme, the phosphoglucomutase. Okay, this is the enzyme that acts on the glucose 6-phosphate, and it gives us the molecule, the glucose 1-phosphate. So it transfers the phosphate group from the 6th position to position 1, and we have the glucose 1-phosphate. Then we have this enzyme which is called the uridine diphosphate glucose pyrophosphorylase. So this enzyme, it's also an enzyme that will act on glucose 1-phosphate and produce what is called the uridine diphosphate glucose. Okay, and this will be our starting molecule in the uronic acid pathway. So in this case, you have the UTP, this is the uridine triphosphate, that will be reduced, or these 2 phosphate molecules will be removed, and only 1 phosphate will remain. And this is the molecule that will have the uridine diphosphate glucose. So this process, the UDP glucose, there will be a dehydration reaction where the water will be removed, and in this reaction there will be an enzyme, the UDP glucose dehydrogenase, okay, in the presence of NAD as a cofactor. Okay, because we know that NAD is the one that will accept the electrons. Okay, NAD, it's involved in reduction reactions, and this going to accept the electrons. Okay, so this reaction that will be catalyzed by UDP glucose dehydrogenase will remove, will produce what is called the UDP glucuronate. Okay, this is the compound that we are going to look at. Okay, and from this compound we go on and have the glucuronate. Okay, where the UDP is removed, we have the glucuronate, and this will go on and produce the L-ascorbate, which is the precursor of ascorbic acid, which is vitamin C. Okay, in those organisms that have the enzyme which is called the glucuronolactone, which I'll talk about later on when we talk about when we look at this pathway in details. Okay, there are some enzymes here that we'll talk about, and also you'll be able to understand why this process coming down here, it can't happen in humans, but in humans we'll go to this process where there will be the production of glucuronides, proteoglycans, and the glycosaminoglycans. Okay, this is just the brief summary of the pathway and we will look at it in details, as in step by step. Okay. So the first step, what happens in this pathway, this is where we have the glucose 6-phosphate, which is first converted to glucose 1-phosphate, and this reaction is catalyzed by the enzyme phosphoglucosamidase. Okay, that's the first step. From the glucose 1-phosphate, this is now where the glucose 1-phosphate will be converted to UDP, which is uridine diphosphate glucose, okay, by the action of an enzyme which is called the UDP glucose pyrophosphorylase. Okay, that's the enzyme that will catalyze this reaction, the glucose 1-phosphate to UDP glucose. Then these all steps that are happening until up to this stage, they are the same as in glycogenesis. Then from there, we have now these other reactions that will follow. This is now where we have the uridine diphosphate glucose dehydrogenase, which is the enzyme in the presence of NAD that will oxidize now the UDP glucose at position number 6. Okay. So the oxidation takes place at position number 6, at carbon number 6. Then this UDP glucose will be converted to UDP glucuronate. Okay, after an oxidation reaction that is catalyzed by UDP glucose dehydrogenase in the presence of NAD as the cofactor. This NAD is the one that is receiving the electrons. It's acting as a recipient of the electrons in this oxidation reaction. So this UDP glucuronate now that we have, this is a metabolically active form of glucuronate. Okay, and this is the one which is utilized from the conjugation of many substances like bilirubin, steroids, hormones, and the other drugs. Okay, so like I said, this molecule or this compound, the UDP glucuronate, it's highly polar. Okay, so meaning it's able to take place in conjugation reaction where it combines with other substances, okay, and make them also highly polar. Once these substances have been made highly polar, then they will be able to be dissolved. They combine with water and they can be transported to the kidneys and be excreted. Okay, so even in these other drugs and other hormones, once they are conjugated, they will become highly polar and they can be, their solubility increases and they can dissolve in water, and it's easy for these substances to be transported. So the UDP glucuronate, this is also required for the synthesis of glycosaminoglycans and also the proteoglycans. Okay, this is a combination of the glycosyl group of carbohydrates in a disaccharide with some amino acids and also some other compounds. Okay, where these other compounds can combine and form one molecule. So this is where this UDP is also required, okay, in these other processes, in the formation of glycosaminoglycans and also the proteoglycans. So we continue with the steps in this UAP pathway. So there is the conversion of the UDP now, glucuronate, to the L-gluconate. Okay, so the UDP glucuronate, after being oxidized to this, will be converted now to glucuronate. Okay, and this in this process, the UDP glucuronate loses its UDP moiety in an hydrolytic reaction, and this reaction it's catalyzed by uh, glucuronidase and releases D glucuronate, which is then reduced to L-glucuronate by a NAD uh, reaction that is also catalyzed by L-uronic dehydrogenase. Sorry, let me just answer this call. Sambas, Sambas. Hey, big man. Hey, okay. Okay, let's continue. So here we have the conversion of UDP glucuronate to L-glucuronate. Okay, so in this case, this UDP will be removed from this glucuronate. Okay, and what will be formed is what is called the L-glucuronate. Okay, so the UDP glucuronate loses its UDP moiety. Okay, once it loses its UDP moiety, then it goes on and forms what is called the D glucuronate. Okay, so this reaction is catalyzed by glucuronidase. This is the enzyme that catalyzes the removal of this UDP moiety from the glucuronate. Okay, and this is an hydrolytic reaction. Then once the D glucuronate is produced, this D glucuronate will be further reduced, okay, to L-glucuronate. Okay, this D glucuronate is going to be reduced to L-glucuronate, and this is by a NAD reaction that is characterized by the L-uronic dehydrogenase. Okay, so the NAD pH is the cofactor in this reaction where D glucuronate is reduced to L-glucuronate. Okay, and the enzyme that is involved is the L-uronic acid dehydrogenase. That's the enzyme that is involved in this reduction reaction. So this is a diagram of just what I've talked about so far. So we have the glucose 6-phosphate. Okay, so this glucose 6-phosphate will be reacted upon by this enzyme, the phosphoglucosamidase, and what will be produced is the glucose 1-phosphate. Okay, this is what will be produced from the glucose 6-phosphate. We can see that there is just a change in the position of the phosphate group from position 6 to position 1. Okay, by this enzyme, the phosphoglucosamidase. Then we go to the next reaction where we have the UDP glucose pyrophosphorylase as the enzyme that will act on the glucose 1-phosphate and attaches a UDP moiety on this glucose. Okay, in the presence of the UDP uridine triphosphate, these two phosphate groups are removed and we remain with the UDP glucose molecule. Okay, so this molecule that we have now, the UDP glucose, this is now where the enzyme, the UDP glucose dehydrogenase comes in this oxidation reaction, and this will now take place in the presence of NAD. Okay, sorry, just a second, I answer this call again. Just a second. Hello? Yes, sir. Mabomba pe wino? Wino. We. Nimpora so apa, maybe not in class with the IDs. With the IDs, uronic acid. I'm taking them on uronic acid today. Yes, you can complete lipids, so that from this one maybe I can go on and do the TCS if I have not done it. You finish lipids. Can you go on maybe some hormones? Hey. From lipids you can go on fatty acids. I don't know, fatty acids or triglycerides, those things. Okay, just check then. Let me know which ones you. Okay, let me just finish this lecture. Then I'll tell you which one I'll do next and which one you can take them through. All right, thank you. Okay, sorry. Let's continue. Okay, are we there, everyone?
Yes, sir.
Okay, that's good.
Yes, sir.
Okay. Let me just get my pointer. Okay, so I'm on this pathway on this diagram. So I ended on this UDP glucose. So we have this glucose which has this UDP attached to it. So this UDP glucose will be oxidized by this enzyme, the UDP glucose dehydrogenase, in the presence of NAD as the cofactor, and the production of hydrogen and also 2 NADH. And what will be released is the UDP glucuronate. Okay, that's a product that we are going to have. Then there will be a dehydration reaction, and the enzymes that will be involved are these glucuronidase, and in this case, the UDP moiety, this UDP will be removed from the glucuronate. And in this dehydration reaction, the UDP is removed, and what will be produced is the D-glucuronate. Okay, that's the molecule that we are going to have, the D-glucuronate. So from this D-glucuronate, there will be the glucuronides, the enzymes that will act on this D-glucuronate, and we will have the L-gluconate that will be produced, and we have the cofactor, then add pH and hydrogen that will give us the NADP. So from this L and from these glucuronides, there will be a series of reactions now that we will look at now further down in the pathway. Okay, so now we look at further down in the pathway where we want to find now the fate of this gluconic acid, the L-gluconate. This L-gluconate, that's the L-gluconic acid. Okay, the L-gluconate, that's the L-gluconic acid. So its fate now, want to see now where it goes now from this stage. So the L-gluconic acid, it has different fates. Okay, in different organisms. Okay, and this is where I said depending on the availability of the enzyme, which is called the granolactone oxidase, which is absent in certain organisms or mammals or animals. Like in humans, we lack that enzyme. So the fate of this gulonic acid will be different, okay, depending on the availability of the granolactone oxidase. So, in the synthesis of ascorbic acid, which is vitamin C, so the L-gulonic acid, this is the direct precursor of ascorbic acid, okay. This is where ascorbic acid comes from. Direct comes from this L-gulonic acid, okay. So in those animals which are capable of synthesizing this vitamin, they are supposed to have this enzyme, the L-granolactone oxidase, okay. All the mammals that are supposed to synthesize this vitamin, they are supposed to have this enzyme, L-granolactone oxidase, okay. So the synthesis of vitamin C, which is ascorbic acid, takes place in these animal species, those that have this enzyme, L-granolactone oxidase, and this is where the fate goes for granolactone in those gunolate in those animals or organisms that has this granolactone oxidase. Okay, so the granulate, it's in a dehydration reaction. It comes to L-granolactone, and this is where we have this L-granolactone oxidase that blocks or catalyzes this reaction. So if this enzyme is lacking, then there will be no production of ascorbic acid or either vitamin C. Okay, so the L-granolactone will be converted to 2-keto L-granolactone in the presence of this enzyme L-granolactone oxidase, and the 2-keto L-granolactone, it's the one that where ascorbic acid comes from. So this process doesn't take place in humans because we don't have this enzyme, the L-granolactone oxidase, okay, but it takes place in other organisms and other animals, okay. So as on this fate, we'll still look at the fate of this uronic acid or L-gluconate, where it's going to be diverted into doing other things like in the production of protoglycans and glycoamino, the protoglycans and those other glycoaminoglycans. Okay, that's where we focus when it comes to humans and the fate of uronic acid. So in a man, that's the fate of uronic acid in humans and other primates, as well as the guinea pigs. Ascorbic acid cannot be synthesized. Okay, in short, it means vitamin C, we cannot synthesize vitamin C in our bodies, but we can take it as a supplement. This is because we lack this enzyme, the L-granolactone oxidase, which converts gluconate to ascorbic acid, and this is because the enzyme is absent, okay, in humans. So vitamin C, we can only take it as a supplement through diet or just being given. So the L-glonic acid in humans and other animals that doesn't have the L-granulation oxidase, this one will be oxidized to 3-keto-gluconate, okay? And the enzyme that is involved is the L-gluconate dehydrogenase, okay, which oxidizes the L-glonic acid to 3-keto-gluconate. This is then decarboxylated to the pentose L-xylulose by the enzyme, the 3-keto-gluconate decarboxylase, okay, meaning one carbon atom will be removed, okay, decarboxylation. So when you talk about this decarboxylation, this is where now you find to know that the pentose sugars, these are a 5-carbon sugar, okay, as compared to when it was coming from the glucose 6-phosphate, it was a 6-carbon sugar, okay. But when it comes down, it's coming down to become a pentose sugar, which is a 5-carbon sugar. This is because at this stage now, there will be a decarboxylation, okay, the removal of one carbon, okay, and what will be produced is carbon dioxide and a pentose sugar, which is the pentose L-xylulose, okay. The enzyme involved is a 3-keto-gluconate decarboxylase. Then this L-xylulose, this is further converted to a xylitol, okay, by an enzyme which is called the xylitol dehydrogenase. And in this reaction, this is a reduction reaction, and this reduction reaction again, it will be followed by an oxidation reaction, okay. This oxidation reaction will now further convert the xylitol to a D-xylulose, okay. We add an L-xylulose, but here this enzyme that will follow will convert this xylitol to the D-xylulose, and the enzyme that is involved is the D-xylulose reductase, okay. It converts the xylitol to D-xylulose via the enzyme, the D-xylulose reductase. So the D-xylulose that we have now, this is the molecule that can enter the exos monophosphate shunt and go down in the exos monophosphate shunt pathway, okay. That's where this one will go, okay. It's going to go and join the exos monophosphate shunt. If you go to the notes, go and check in the exos monophosphate shunt, you'll be able to know where the D-xylulose joins and what it does that side, okay. And I will expect you to know that, okay. Go to the exos monophosphate shunt, go and check, look at the D-xylulose and see where this D-xylulose is joining that exos monophosphate shunt, okay. So the D-xylulose is converted to the xylulose 5-phosphate via the enzyme xylulokinase, okay. So this xylulokinase is usually initially just adding a phosphate group at position 5 of the xylulose sugar, okay. The xylulokinase, it's adding a phosphate group at position 5, okay. It's phosphorylated at position 5. So the xylulose 5-phosphate can enter the exos monophosphate shunt and continue in that process, okay. So when you go back to the notes in the exos monophosphate shunt, you'll be able to see where we we have the xylulose 5-phosphate. Okay, and what it does that side. So this is our table of which is just a summary of what we've been talking about. We have the L-gluronate, which is the L-uronic acid. Here, it goes and produces ascorbic acid on this side, okay, in those organisms that have the L-gluronolactone oxidase, okay. So those ones that have this enzyme, which as humans doesn't have this enzyme, which is here. This is the fate of L-gluronate. Then for those that doesn't have this enzyme, the L-gluronate comes down here, okay, where we have the 3-keto L-gluronate, okay, in those reactions that I'm just from talking about. So we have the 3-keto L-gluronate, we have the L-xylulose, that's after a decarboxylation, okay, the 3-keto L-gluronate being decarboxylated by the 3-keto L-gluronate carboxylase. We have the L-xylulose, which is the pentose sugar, and under the action of the enzyme, the xylitol dehydrogenase, in the presence of pH and hydrogen, we have the xylitol, okay, and this xylitol will go on again through the action of the xylitol, the D-xylulose reductase enzyme, which is here. Then we'll produce the D-xylulose, okay, and this D-xylulose will be acted upon by the xylulokinase, which is the enzyme which is here in between. And what we are going to have, it's the xylulose 5-phosphate. And this will go on and join the exos monophosphate shunt. Okay. That's in animals that have no gluronolactone oxidase. That's the fate of the uronic acid. Okay. In those animals that doesn't have this enzyme, but those that have the gluronolactone oxidase, the gluronate will go on and produce vitamin C, which is ascorbic acid. So this diagram is the same as the one that I'm just from talking about, just that it's indicating everything starting from the glucose 6-phosphate until the end. Okay. So we look at the importance of this uronic acid pathway. Okay. So the importance of this, like I said when we are starting this topic, it's highly polar. Okay. It's highly polar, and that's why it's very, very important when it comes to conjugation of certain substances in the body. So the UDP, uronic acid, which is the active form of uronic acid, it acts in the conjugation of bilirubin, in the conjugation of steroids, also in the conjugation of various drugs, which will make them more water soluble, and this will make it very easy for these substances to be excreted from our bodies. Okay, and this uronic acid is also important in the synthesis of glycosaminoglycans, the GAD. Okay. It's very important in the synthesis of the glycosaminoglycans. The glycosaminoglycans, like the proteoglycans, these are very important molecules in the extracellular matrix. Okay, in the extracellular matrix, you are able to find these. They play a number of roles in the extracellular matrix. Okay, starting from the stability of the extracellular matrix, make it more stable, and different functions. Okay, glycosaminoglycans, including the proteoglycans. So this also has an effect, okay, on effect of drugs on uronic acid pathway. Certain drugs, they have an effect on this uronic acid pathway, and these drugs, we have an example of the barbital, aminopyrin, antipyrin, the chlorobutanol. These significantly increases the uronic acid pathway to achieve more synthesis of uronate from glucose. Okay, and this will lead to the availability of more glucuronate for conjugation purposes. Okay, so more of glucuronate will be needed. Okay, so that the bilirubins can be conjugated, the steroids and other hormones. Then certain drugs were found to enhance the synthesis of ascorbic acid AD. Right. Okay. So once certain drugs are given, they help in the synthesis of vitamin C, and we know vitamin C, it has different functions in the bodies. Okay, a lot actually. Have we done the vitamins? Harry?
No, we have not.
You have not done the
We've not done
vitamins?
vitamins. No.
Okay, I think we should do it next. I have not done. We need to do that one. We're supposed to do it, I think, even before we started these other pathways. We'll see what we can do. We can do it next. Okay, I think the uronic acid pathway, this is where it ends. It's not that long. I want where you are confused, where I can, any questions where you are confused, where I can tackle, where you are not clear about this pathway.
Sir, just a question, though I joined in late.
Okay,
Yes.
you can go ahead with the question. Yes, madam. We are waiting for the question.
I'm asking, are we going to have a recording of this lecture? Because
Yes, I'm recording, so you are going to have it.
okay, thank you.
You're going to have it. And how many are we? We didn't even see how many are missed, so that we see where we are.
We are 7.
Oh, okay, not bad. Let's see half. Any other question? Okay, so there are no further questions. Mr. Kizito, he called me just a few minutes ago. I think he will meet you. I just want to, once I'm done, to discuss which topic he can tackle, so that we move, because we are still very much behind, but we move. So I'm sure he will communicate with you, Alec.
All right,
And concerning the makeup test, you also communicate once he's ready, then we can do it. So for
all right,
the
thank you.
makeup test, it's everyone can attempt, but for those, if you passed, it's either you attempt, you forfeit one. If you if you attempt, it means you forfeited the other one. That's how it is. So if you attempt and get less marks in the makeup test, then it means if you had the 50 in the first test, you can't come and say, no, I want to have that other one. No. So it's only those who feel they want to attempt. They are the ones who should attempt. Okay.
Okay, sir, noted.
Okay. But if you have any questions, even outside our lectures, you can still send me. Okay. You can send me even an email. I prefer the emails, anything to do with school. You can send an email, especially if you have questions. Okay, you guys, it's distance and we are not together most of the time. So if you have any questions, it's better you send me even via an email or even on WhatsApp. I can still get back to you. Yeah. If you have any questions, make sure you communicate. Send me that question. I'll look at it and respond to you. But don't just pack these notes. Okay, it's a problem if you pack these notes. From here, find time, go through and try to understand the pathway properly. Okay, you understand it so that you see how is it flowing. You need to understand it. But if you go and pack these notes, you are not going to know anything about it, and you won't be able to answer anything about it. But make sure you don't pack these notes. Go through the handout, try to understand it. If you have one or two questions, you can still come back to me and ask where you are not clear. But go through the handout, understand it. You can even go through the books, read the books. I send you we communicated the books which are recommended. You can go through the books. Although in the books it's bulky, but you can try and see how far you can go. Okay. But with these notes, these are summarized. Go through the notes, don't pack them. If you pack them, definitely you are going to forget. Because these pathways you need to go through. You go through frequently so that it's just there. You are able to master it. You master these pathways, because these pathways you can't memorize. You need to understand them. You need to know. Okay. Okay, if there are no questions, I think we will end here, since people need to